Everybody. Welcome to our monthly scientific oncofertility meeting. My name is Ariella Shikanov. I'm an associate professor in biomedical engineering at the University of Michigan. And today we have the amazing opportunity to welcome Dr. Ewelina Bolcun-Filas. I have a small introduction to tell you all about magnificent Ewelina. So she was born in Poland, and she obtained her master in biology in Jagiellonian University in the Department of Genetics and Evolution in Krakow, in Poland. After earning her PhD in Human Genetics Institute at the Georg August University in Germany, she pursued her interest in reproductive biology and genetic causes of infertility in Dr. Howard Koch lab as a postdoctoral fellow at the MRC Human Genetics Unit in Edinburgh, in Scotland. And then there, she investigated the process of meiotic recombination during sperm and egg development. In 2008, she moved across the ocean to reunite with her family in the United States and joined Dr. John Shimedi's lab at Cornell University for further study of genetic causes of infertility. She joined the Jackson Laboratory, where she is an associate professor right now, in January 2015, and she continues her work in genetic causes of infertility and the role of CHEK2 pathway in cancer treatment toxicology. The most exciting event, I think, she deserves congratulations on publishing a beautiful paper last year in Science Advances, and the title of the paper is CHEK Signaling is the Key Regulator of Oocyte Survival After Chemotherapy, and she will be talking about this today. I certainly hope so. And just last sentence, Evelyn is funded very nicely. She's funded by Bill and Melinda Gates Foundation, and also she is a participant of multiple NIH-funded center grants. So with this, Ewelina, the screen is yours.
All right. Can you see my pointer?
Yes, perfect.
Perfect. Thank you for the introduction, very generous. But it's really my pleasure to be here and tell you about research in our lab that made us consider CHEK2 as a target for ovarioprotective treatment. And we envision CHEK2 inhibition as a way to prevent follicle depletion and this way to extend ovarian function in cancer survivors. But we also recognize that CHEK2 is a checkpoint protein and involved in DNA damage repair. So we also have to weigh the benefits and potential risks of targeting CHEK2. I hope most of you had a chance to see Carla Hutt's presentation last webinar, because she gave a fabulous introduction to the issue of cancer treatment toxicity and its effect on the reproductive system. So I'm not gonna mention this today. I really highly encourage you to see that. I want to start with emphasizing that we are getting much better now with cancer diagnosis and cancer treatment, which means cancer patients now survive much longer, and we have to kind of switch gears and think more about the quality of survivorship, and that includes reproductive health. So it's estimated now that there are about 18 million cancer survivors living in the US, and more than half of them are women. But with this improved survivor and longer life, we are now observing many long-term and late onset health problems that affect survivors' health. And they are caused by these life-saving, but often cytotoxic treatments. And these late side effects depend on the type of treatment, patient's age, and probably a few other factors. And they may not show up for months or years after treatment, especially some of the reproductive issue. And what we are hearing more about in terms of cancer toxicities and side effects is hearing loss, cardiotoxicity and cardiovascular problems, chronic pain and neuropathy, kidney failure. But and thanks to Oncofertility Consortium, now we also learn more about reproductive health issues. And it's not restricted to infertility. It also means that patients can experience premature menopause earlier in life, problems related to menopause, as well as sexual dysfunction. So all of these long-term health problems can have significant impact on the quality of survivorship and have both financial and psychological burden. And when you think about reproductive health, IVF procedures and fertility preservation are costly, and they are not always covered by insurance, and inability to have children can also have psychological or emotional burden. So altogether, we really have to think about how to help cancer survivors to have a healthy and fulfilling life. And when we think about toxicities of cancer drugs on the ovaries, I really want to emphasize that healthy ovaries are not only important for women's reproductive health, they're also important for women's overall health. The follicles in the ovary that are sensitive to some of those cancer treatments have two major functions. One function is to grow and ovulate eggs, which are needed for fertility. But growing follicles in the ovary produce hormones, estrogen, and after ovulation produce progesterone. And these hormones are important for puberty onset in girls, maintenance of pregnancy, but also they support cognitive health, cardiovascular, and bone health in women. So once those follicles are depleted, which can be accelerated by cancer treatments, ovaries cease producing hormones, and that triggers the onset of menopause. And I think everyone knows that menopause also increases the risk of other diseases. And that includes dementia, Alzheimer's disease, stroke, heart attack, and osteoporosis. So really, when we think about reproductive health, we should also take that into account, that we want to not only preserve the reproductive function of the ovary, we also want to preserve the endocrine function of the ovary. Luckily, there are really good fertility preservation options for women. Embryo, egg, and ovarian tissue cryopreservations among them are the most successful. However, some of them require delays in cancer treatments, embryo and egg freezing, because they require retrieval of eggs. For embryo freezing, women require partners, so it may be limiting for younger women or single women. And these two options are not feasible also for prepubertal girls, because they don't yet respond to hormones. And for them, ovarian tissue cryopreservation and later in life grafting is now the best option. And it has been done around the world successfully, which is great news. It's not so good on the front of preserving the ovarian function. Actually, the methods to ensure that ovaries will work long-term are quite limited. The current treatment options include ovarian suppression using GnRH agonists, which is mostly used for breast cancer patients when there is a need to decrease production of estrogen, and it has been shown to successfully delay menopause in these patients. But the data on effectiveness of this option for other cancers are mixed. Again, I'm not an expert. This is basically based on what I read in the literature. So we need some improvements here as well. For patients who undergo radiation, especially the pelvic area, radiation shielding or surgical repositioning of ovaries out of harm's way can be used. But this is limited, as I said, to radiation, and also has variable results, and sometimes will also need a surgical intervention. And although ovarian tissue freezing, I think, is mostly used for preserving fertility, it can also sort of help in recovering the ovarian function, as these grafts can last longer. And I think the lifespan has been shown to be from few months to years, and maybe 7 years was the longest that I found. But it also depends on follicle density and size of the graft. And sometimes continuous function of the ovary would require multiple grafts, which again involves substantial financial investment and multiple surgeries. So really, we don't have a lot of options to actually help to preserve ovarian function. I would say that there are some good options on the horizon that many labs are working on. We know that ovaries can be transplanted and they can function, but I think we have to improve the way whole ovaries are cryopreserved. and later transplanted. Bioprosthetic ovaries, I think that's an exciting avenue that probably is kind of far away still, but has been shown to work in mouse and successfully produce offspring. Stem cell derived germ cells, again, I think it's quite far in the future, but we could envision these stem cell derived oocytes to be used in those prosthetic ovaries, for example. I think what's kind of a low-hanging fruit now and maybe more achievable is the pharmacological inhibition of follicle loss. And there are many labs looking into that in different animal models. And so far, I think sphingosine phosphate and its agonist FTY720 have been shown to preserve ovaries after treatments, not only in mice, but also in primates, rapamycin as well, quercetin, and few other molecules have been shown to actually improve ovarian function after cancer drug toxicities. What I'm going to focus today is our work on the CHEK2 as one of those pharmacological options. So first, I would like to talk about the potential benefits of CHEK2 inhibition for the reproductive health of cancer patients. So why CHEK2? I think there are multiple good reasons why CHEK2 could be a good, what I would call ovarioprotective treatment. First, obviously, is our work where we showed that CHEK2 deficiency prevents elimination of primordial follicles after radiation and chemotherapy. We've showed this in mice, but I think there's a reason to believe it will be the same in human. A large GWAS study from 2021 also linked CHEK2 loss of function alleles to delayed menopause in women. And this was a large study that found out that CHEK2 loss of function alleles are good predictors of later onset of menopause by about 3.5 years. They also showed that BRCA mutations, or loss of function alleles, are predicting earlier onset of menopause by 1.5 or 2.6 years earlier. They also found and confirmed few known hits as well as a new one. A really nice study. I encourage you to read that. In the same study, they also showed that CHEK2 deficient mice have a larger and longer lasting ovarian reserve. That's something that we have observed in our earlier study. But at the time that we looked at, sort of around puberty, we could see a trend, but it wasn't significant when we looked at a larger number of animals. What they did, they looked further, so they aged mice to about 13.5 months, which I think in human years would be about 50 years, so around the time of menopause. And what they could see is that in CHEK2 deficient mice, here in white boxes, you see that their primordial follicles, as well as growing follicles, are larger in numbers, and this difference is significant. So when looking at earlier ovaries, where there is a trend that is higher later in life at 13.5, it suggests that maybe CHEK2 deficient mice are born with slightly larger ovarian reserves, but really the difference is throughout a female's life, where it seems that they are not losing as many follicles throughout their life. They aged these mice even further to 16 and 17 months, which I think would be about 60 years old in human years, and looked at the concentration of AMH hormones, which is the indicative of how many follicles are growing in ovaries. And again, they could see a significant or significantly higher level of AMH in CHEK2 deficient mice. So this all indicates that CHEK2, or confirms that CHEK2 is important for survival of oocytes, and that interfering with CHEK2 function can potentially improve follicle survival in human. I believe that pictures are worth 1,000 words, so I'd like to show you how radiation damages ovaries in the mouse. I think the 3D images are more impactful than sections. And here what you see is a mouse ovary that was immunostained with marker for oocytes, DDX4, here in white, and you can see a 3D rendering of the whole ovary. What you will see on the bottom are these sort of donut-shaped cells, which are the oocytes in primordial follicles, and the larger, brighter oocytes, which are oocytes in growing follicles. When we expose wild-type mice to radiation, and this is actually relatively low dose, 0.5 gray, you can see that within 2 weeks, all of those primordial follicles are gone. You see sort of void in the place where we should see them. And only the larger follicles survive, which actually don't undergo apoptosis. They sort of keep growing and undergo atresia later. I also want to mention that mouse oocytes seem to be exquisitely sensitive to DNA damage. So really, with these treatments, we may be modeling the most severe types of ovarian toxicity. And just as a side note, in mouse, we use a dose of 5 to 6 grays to ablate hematopoietic stem cells in the bone marrow. So you can see that this is quite low compared to that. Now, when we expose CHEK2 deficient mice to the same dose of radiation, we see abundance of primordial follicles surviving in those ovaries. And we can't really see a difference between untreated and treated ovaries. And those follicles and oocytes not only survive, they are fully functional, they can recover from DNA damage, and they are producing healthy PAPs. So we didn't really see differences in the number of offspring born from CHEK2 untreated and treated females. And these pups aged to, I think, at least 1 year, they did not see spontaneous cancers or really any problems later in life. In contrast, the wild-type females exposed to this dose of radiation never recovered from infertility. So I hope this illustrates how powerful CHEK2 inhibition could be. It could really dramatically improve survival of primordial follicles. So initially we were concerned that maybe it's specific just to radiation. So later we followed up to see whether CHEK2 inhibition or deficiency could protect follicles after other types of genotoxic treatments, for example, different types of chemotherapies. So here we are switching gears to explant culture that allows us sort of better control over drug treatments, dose, and timing. And through these cultures, ovaries kind of become flattened, more like a pancake. That's why you see they are sort of more spread out than the in vivo ovaries. So we chose 4 different treatments for testing mafosfamide, which is an analog of cyclophosphamide that we can use in culture, cisplatin, and 2 topoisomerase inhibitors, doxorubicin and etoposide. And these 2 in human seem to have lower ovariotoxic effects. But at least in mouse, we see that doxorubicin is, even at low levels, is toxic to primordial follicles. And the same we see for cisplatin and mafosfamide, where primordial follicles are almost completely or completely depleted. For etoposide, we do see lower toxicity in the wild type. So again, we wanted to see whether CHEK2 deficient ovaries will do better. And indeed, we see improved primordial follicle or oocytes in primordial follicle survival. And they are marked by magenta color for P63. And they are nicely seen here that in all cases, for all drugs, CHEK2 deficiency prevents elimination of primordial follicles. You can also see quantification here. So again, altogether, this told us that CHEK2 is powerful for prevention of follicle elimination during these toxic treatments. We wanted to be sure and did that later also in vivo. And we picked cyclophosphamide and cisplatin as the 2 sort of most ovariotoxic treatments. And we injected juvenile mice with a single acute dose of these drugs. that has been used by many of our colleagues to demonstrate toxicity. And in our control CHEK2 heterozygous mouse, we see that after cyclophosphamide treatment, we see elimination of primordial follicles. It may not be super visible, but here you can look at the numbers. We mostly observe sort of empty follicles on the edge of the ovary and some defects in granulosa cell proliferation. For cisplatin, we don't see a complete depletion as we see with cyclophosphamide, but these numbers are severely reduced. We see still primordial follicles as well as sort of abnormal growing follicles. But what we've seen in CHEK2 deficient mice confirmed what we've seen in ex vivo. They still have abundant population of primordial follicles even after treatment with cyclophosphamide and cisplatin. And I hope you can see these follicles here. And again, this is all quantified here on the right and indicates again that CHEK2 deficiency can prevent elimination of primordial follicles after in vivo treatment. What we also observed was that CHEK2 deficient mice after treatment with cyclophosphamide recover slightly better than the control animals that kept losing weight. CHEK2 deficient mice sort of kept gaining weight, not enough actually to keep them for fertility tests. We decided to not to breed them, and I think we just started with too high dose of cyclophosphamide to kind of that induced a lot of other health problems in these pups. Nevertheless, even with this dose of cyclophosphamide, we could see abundant follicles. And from other studies, we know that if follicles survive, most likely they will be functional. So all of this together made us think that CHEK2 sort of offers really good therapeutic opportunity for what I would call ovarioprotective treatment. And I sort of want to differentiate between fertility preservation and ovarioprotection, because I think the consequence of this treatment may sort of limit the use of inhibition for reproduction. But generally, we think that CHEK2 inhibition could protect ovarian follicles during the duration of chemo and radiation treatment, sort of at the same time, sort of more of a short-term treatment. And this could prevent or delay premature loss of ovarian endocrine function. And as a consequence, it would also prevent infertility, but it does require more studies to ensure germline genome integrity. So we wanted to test if a pharmacological inhibition of CHEK2 would do the same. So we started looking for inhibitors, and I was actually surprised that there aren't as many CHEK2 selective inhibitors available. Many of them can inhibit both CHEK1 and similar kinase, CHEK2 and similar kinase CHEK1. Nevertheless, we found this one, CHEK2 inhibitor 2, which I think is also known as BML277, and decided to test it in our ex vivo system. And what we observed through that is that with increasing doses of the inhibitor, we could see increasing survival of oocytes after treatment with radiation. And again, you can see here these small oocytes, and now in green, and DDX4 or MVH in red. The survival was not as great as with the knockout, but we transplanted these treated ovaries ex vivo into the bursa of mouse that were infertile. And what we got were pups that were born. These pups were healthy. And in contrast, we did not get any pups from ovaries that were treated with radiation, but without the inhibitor. This inhibitor was later used by Soo-Yoon Kim to show that it can also improve reproductive outcomes in mice treated with cisplatin, and she did that in vivo. And that sort of confirms that CHEK2 could be used also in vivo for protection of follicles. As I mentioned, we weren't really happy with the survival, so we started looking for other inhibitors, and there aren't really that many. So we selected 4 that we wanted to test. Only one of them had higher selectivity towards CHEK2. The other ones are sort of known as dual CHEK inhibitors. They can inhibit CHEK1 and CHEK2. And what was intriguing is that these CHEK2, dual CHEK2 inhibitors have been shown to sensitize cancer cells to chemotherapy and radiation. So when we think about this, inhibition with those dual inhibitors could achieve 2 things. It could enhance cancer treatment as well as protect ovarian follicles during cancer treatment. So sort of a treatment that would have dual benefits as well. So we wanted to test these inhibitors. And again, we chose ex vivo system because it's easier to control the doses and treatments. And I used them first with radiation. And we were kind of disappointed, very disappointed, because 3 out of 4 did not show any improvement with increasing doses. And for some of them, we tried higher and it did not improve. Indicating that they don't sufficiently inhibit CHEK2 in our assay. With the exception of AZD7762, which showed increased survival with the increasing dose, and we were able to observe plenty of primordial follicles sort of on the edge of this ovary. So that was exciting. At least one of them showed beneficial effects. And again, we wanted to confirm if that would work with chemotherapy treatments. So we treated ovaries with the 2 drugs I showed you before, mafosfamide and cisplatin. And hopefully you can see that without AZD, there are almost no primordial follicles left in the ovary, and you see plenty in the control. However, in ovaries that were co-cultured with mafosfamide and AZD or cisplatin with AZD, we actually see a really nice survival of primordial follicles, and they looked healthy. And here you'll see quantification. There was sort of dose-dependent reduction actually in survival. And for those of you who have really good eyes, you can probably notice that these ovaries are much smaller. We found that AZD treatment kind of retarded growth of those explants compared to untreated or even compared to drug-treated. And you also probably observed that the oocytes in growing follicles, they kind of stuck on top of each other, while in sort of healthier ovaries, we see them separated. And this is because there are at least 2 layers of granulosa cells around them that we are not visualizing. So this kind of retarded growth made us think that even though primordial follicles do survive, there is a problem with proliferation and most likely of granulosa cells through potentially AZD inhibition of CHEK1. So we decided to test this and looked at DNA damage and tunnel staining for apoptosis. And indeed, we found that even without radiation, AZD treatment induces high levels of apoptosis in ovaries, which is even higher after radiation. But we do observe follicle survival, and those are healthy. They don't show any apoptosis, in contrast to just irradiated oocytes that are apoptotic. So you can pick a few apoptotic oocytes here. So that was bad news, because it meant that although this inhibitor could protect oocytes through inhibition of CHEK2, it has detrimental effects to proliferating cells. Later, we also found that this inhibitor was in clinical trial for synthetic therapy, and that trial was discontinued because of cardiotoxicity in a trial subject. So this one is a no-go, but it doesn't mean that a different type of inhibitor could be beneficial for oocytes without affecting granulosa cells. But we still wanted to confirm that AZD inhibits oocyte loss through inhibition of CHEK2. So we used Western blot to look at phosphorylation of direct target of CHEK2, P63, which we showed before, that is activated by CHEK2. So through in vivo and ex vivo treatment, you see that P63 shows this mobility shift as a result of phosphorylation. But after treatment with AZD, we don't see this shift, which means that P63 is not phosphorylated, which looks exactly the same as what we see in CHEK2 deficient mice. You can also see that P53 is activated by radiation, and also P53 is activity is decreased by AZD. Again, confirming that AZD inhibits CHEK2 and CHEK1 as well, because CHEK1 can sometimes supplement in P53 activation. We also see accumulation of phospho-CHEK1, so the active version of CHEK1, which is non-functional, we think, and this is a result of abundant DNA damage that we see in AZD-treated ovaries. So taking all of this together, we can conclude that short-term inhibition of CHEK2 is beneficial for primordial follicle survival, but this inhibitor is not good. We need to find highly selective CHEK2 inhibitors to protect follicles and limit cytotoxicity, right? Because we don't want to treat one toxic effect and induce other toxic effects. So there's still a lot to do, but I think that CHEK2 is still a contender for preserving follicles in cancer patients. So let's switch gears now and let's talk about the potential risks. We all know that CHEK2 is a checkpoint protein, and it plays a role in DNA damage response, in particular to double-strand breaks, and within the homologous recombination repair pathway. CHEK2, together with CHEK1 and two apical kinases ATM and ATR, are responsible for cell cycle arrest, first to repair DNA damage, and if damage is not repaired, to induce apoptosis. And you can see a lot of arrows going between different proteins, and indeed there's a lot of crosstalk between these proteins. It would be probably very bad for us if we just had one pathway to do that, so there is some redundancy built into the system. And even in the absence of CHEK2, P53 can be activated by either CHEK1 or ATM kinase as well. Nevertheless, because of CHEK2 involvement in this DNA damage response, we really have to think about the consequences of inhibiting CHEK2. So one worrying sign is that CHEK2 pathogenic variants, which are rare, they have been found to be associated with moderately increased risk of breast and slightly increased risk of colorectal cancer. So we really have to think about this. So for average person, the risk of breast cancer, or for women, the average risk of breast cancer is about 12.5%, and that increases to between 20 to 40 for CHEK2 mutation carriers. So this is something significant. The risk also is doubled for colorectal patients. However, I want to so stress that carriers of CHEK2 mutations, they have sort of lifelong exposure to CHEK2 deficiency, basically from conception to the age of 30, 40 years, where cells were deficient for CHEK2. Where we think about protective treatment, we are thinking more about a temporary treatment that may not come with the same risks. And even for breast cancer patients, CHEK2 is quite rare in cancers, and much more rare than BRCA1 and BRCA2 mutations. However, it's definitely found in breast cancer as well as in a few other cancers, prostate, gastric, pancreatic, and so on. Another thing I would like to sort of mention, and maybe that's for the discussions later, that those pathogenic variants for CHEK2 may have different consequences. Some of them will lead to loss of function for CHEK2. Some of them may be truncating or have a gain of function, which could sort of change how CHEK2 operates. So altogether, this is concerning, and what we need is more studies to test if short-term CHEK2 inhibition could bring about risks of cancer, if it could affect cancer treatment, or increase the risk of cancer relapse, cancer treatment resistance, or appearance of secondary cancer. And altogether, it also may be that not all patients may actually benefit from ovarioprotective inhibitor CHEK2 inhibition, and that should also be taken into account when we think about using CHEK2 as a target for ovarioprotection. Another concern that is often raised by reviewers of my papers and grants is that whether the oocytes that we preserve after damage, they are intact and healthy to be used for reproduction. Because we do know that radiation and chemotherapy induce double-strand breaks in oocytes, which you can see in this image with staining with gamma H2AX. And there are between just a handful to 30, 40 breaks that we can detect in immature oocytes. And those breaks theoretically can become mutagenic. However, multiple studies in mice using mouse knockouts for CHEK2, P63, Puma, and Noxa have shown that inhibition of apoptosis actually allows more time for DNA repair in oocytes, and what those I call rescued oocytes produce healthy offspring, with a caveat that mutations will be still in a heterozygous state, so may not be revealing themselves in this generation. Carla Hatt's lab did a really nice study where they looked at mutation rates, actually sequenced the genomes of the pups after radiation, and they didn't find any significant increase in the mutation rates, which was a good sign. But again, the caveat was that maybe just the best oocyte get to ovulation and allow survival of the pups. I would argue that the risk of mutagenic effects of those double-strand breaks in oocytes are low. And one reason to believe so is that all oocytes predominantly use homologous recombination as a repair pathway, which is a high-fidelity repair that does not induce as many breaks as sort of dirty ligation of broken ends through other repair pathways. Also, DNA damage is introduced at the stage when oocytes are arrested at dictyate. So for every chromatid, there are 3 templates for repair. So that also increases the high fidelity repair. Another aspect I want to mention that I often discuss with my computational colleagues when we talk about sequencing oocytes or PAPs to look for those mutations is that double-strand breaks occur randomly in the genome. Maybe there are some hotspots, but generally they are not in the same spot, in the same gene across different chromatids. So they are introduced at the stage when oocytes are arrested at dictyate, so they are still diploid, but they are after replication, so they have 4 cDNA content. So let's think about 4 chromatids for every chromosome. And when you think about meiosis, which reduces the number of DNA that gets to the egg, the damaged genome that ends up in the ovulated egg is sort of a quarter of what was initially damaged. So when we think about 40 breaks for easy math, if all of them are repaired by mutagenic pathways, 10 of them will end up in the oocyte. And if the mutagenic effects are just 50%, there will be 5 breaks, if my math is correct, right? So this kind of illustrates that intrinsically, because of meiosis, the risk of transmitting all the mutations to the offspring are already decreased by 25% at the onset. Nevertheless, we don't really know how breaks are repaired in human oocytes, so that's something that we have to further investigate. And even if the risk of mutagenesis is relatively low, more studies are still needed to test the safety of CHEK2 inhibition in human or other model organisms. And finally, if we do use CHEK2 inhibition as a way to preserve follicles, it may be still more suitable for preservation of ovarian function, with the understanding that preservation of fertility should be done by other methods, for example, cryopreserving gametes before treatment initiation. So with that in mind, I again want to stress the analysis of risks and benefits of preserving follicles, whether by CHEK2 or by other methods that prevent follicle elimination. So what are the benefits of CHEK2? Is the preservation of primordial follicle pool and improved reproductive, and because of that, overall health of cancer survivors. The potential synergistic interactions between CHEK2 inhibitors and chemotherapy treatments should also be considered, as they could help with cancer treatment. But there are risks that we have to keep in mind. One is that CHEK2 mutations are associated with moderate or low risk of cancer, but we still don't know about risks associated with short-term inhibition. And inhibition of apoptosis in damaged oocytes, at least from animal studies, that not seem to increase the risk of abnormalities or mutations in the offspring. But again, that's something that we'll have to investigate further to ensure that we are not damaging future generations. So I think the bottom line is that I believe it's promising, but we still need more studies to assess the efficacy and safety of CHEK2 inhibition. So with that, I would like to thank Chihiro and Zach, who are driving this project. We are a small lab, and now this project is continued by Monique and Alicia. Before she was a postdoc, I looked at the relationship between genetic diversity and response to cancer treatments, and other lab members who helped with that, CHEK Scientific Services, and support from Cancer Center. And most of this work was funded by a B Foundation grant. And with that, I would like to thank you, and I'm open for questions. All right, and I should stop sharing, right, so I can see you all. All right.
Can I ask a question?
Yes. Hi.
Hi, Ewelina. It was a beautiful talk. I always admire you. It was fantastic, your talk. So I have a question about your CHEK2 study. Do you think it would be okay for normal cell in our body if we use CHEK2 inhibitor? That's my first question. The second question is that, do you think primordial oocyte is special DNA repair mechanism than other mechanism in other cell type? What do you think about it?
Yeah, like CHEK2 is there for a reason, I would say, right? To sort of protect from damage. So it may be difficult to apply CHEK2 inhibition throughout our lives. Although, we have CHEK1 that does a lot of sort of genome guarding. And at least from CHEK2 deficient mice, we don't see increased rates of cancers in them, so they can repair DNA damage without CHEK2. And from the human GWAS studies, again, you could interpret this in different ways, and we don't know about the health of these women. But if they survive to have a longer onset of menopause, it means that they were healthy, and the lack of CHEK2 probably did not impact their health, to the extent that they would shorten their life. So I would say, bypassing safety is never good. But if there aren't too many challenges, I think it may be okay to do it, and maybe okay to do it in a short term if the benefits outweigh the risks. As for DNA repair, I think we still don't understand well how oocytes repair DNA damage. We see this difference between primordial follicle oocyte versus larger oocytes, where they do have p63 CHEK2, but they don't trigger apoptosis. So there's still a lot to discover, and probably maybe discover proteins that could enhance DNA repair rather than block apoptosis. So I don't have a good answer. If there's anything different, there may be, but we just don't understand it well yet. Thank you.
Thank you so much.
Check.
Hey, Ewelina.
Yes.
Hey, very nice work. So for between your ex vivo and in vivo data, do you always see the consistency? Like because there's no pharmacokinetics ex vivo.
Yeah, so that's something to keep in mind also, for a human. And when we think about mice, we use Black6, which generally are quite resistant to DNA damage, but on the end of oocytes, they're kind of very sensitive. We have some other work going on in the lab showing that not all mouse strains react to drugs the same way, which could either be through different pharmacokinetics. For example, for cyclophosphamide, the metabolism of cyclophosphamide in the liver could differ between patients, and how those metabolites are transported to the ovaries and to follicles. So generally, I think we see good consistency between ex vivo and in vivo, but there's sometimes differences because those doses can be difficult to match in media versus what's in the bloodstream and actually gets to the oocytes.
Can I ask one more question? Can you hear me? Okay.
Yeah.
So you showed Western data after treating radiation, you could see P53 and also CHEK1 expression. Do you think oocyte turn on P53 or somatic cell turn on the P53 and CHEK1? What do you think?
Yeah. So yeah. So this was a long journey to actually find P53 antibodies that actually work in mouse in response to treatment and show where it's supposed to be. So what we found is that, yes, P53 is activated in oocytes as well, but it doesn't necessarily induce apoptosis at sort of lower thresholds. I think the relationship between P53, P63, and CHEK2, it's kind of, again, a balance of what type of DNA damage is induced in oocytes and how those two proteins are being activated. So, and like radiation and chemotherapy treatments can induce not only DNA double-strand breaks, right? They can induce oxidative damage, protein damage. So I think there's a lot of triggers that can activate CHEK2, P53, and P63 slightly differently, depending on different drugs and how much damage these drugs are inducing in the oocytes. So it's there in both, in oocytes and in granulosa cells and in other cell types.
Thank you.
Ewelina, one more question
basic
is
science, but
yeah. So one more question is, I noticed you used the global CHEK2 knockout mice, and sorry, I forgot how about the oocyte conditional CHEK2 like a knockout mice. But overall, the preservation of pregranulosa cells in the primordial follicle, is that because the deletion of the CHEK2 in granulosa cells, or just indirectly because the deletion of the CHEK2 in the oocyte? So if the oocyte is preserved, so and then the somatic cells function is preserved too, or both maybe.
yeah, so that's a good question, and it brings the expression of CHEK2 and P53, which are expressed in every cell. versus P63, which is expressed only in oocytes. So we haven't tried to generate a conditional for CHEK2. We did sort of what I call Franken ovaries, where we separated somatic cells and granulosal cells from CHEK2 deficient and wild type mice, and we put them together, and it seems that deleting CHEK2 in oocyte was sufficient for oocyte survival. Again, we haven't followed up on that as much, but or did like quantitative study. But to our knowledge, for now, that inhibiting apoptotic pathway in oocytes seems to be sufficient to rescue majority of oocytes. However, I think that signaling from granulosal cells, I think it's possible, given that they are really, maintaining signaling between them and keeping follicle arrested. Because I see that there are two ways of escaping apoptosis after DNA damage. One way is apoptosis itself, and another way is activation. So if oocytes activates to grow, they sort of, I think they can bypass the apoptosis and sort of keep growing towards atresia, but not undergo survival. And like this activation, how much of it happens really, depending on the treatment, I think it's still open discussion. I don't think that's solely responsible for loss of follicles, but I think it can contribute to follicle loss.
Yeah, that is correct. I'm always like impressed by Soyun's P63 conditional knockout mice in oocyte. It seems whenever the oocyte is protected and then the whole follicle is protected too, they really talk to each other.
It's important to think about that. Sometimes, I get questions about granulosal cells. They are equally important, and they are, providing that there's the oocytes inside, right? So we really need to think about protecting oocytes first, and then finding ways to improve granulosal cell survival to support them.
Yeah, totally agree with you.
Very cool.
Or maybe Alina have another question. So sometimes, we are doing some Doug Rubinstein work too. So how about are you considering to use a cancer cell model, like a cancer model in animals?
Yeah, so that's something that I always consider, and we have very good PDX score at JAGS. So my concern sometimes with using cancer models are whether we can keep the cancer on hold for long enough to see the effects of preservation of ovarian function, right? Because sometimes the cancers can overwhelm the mouse and sort of die earlier. I think that's something that eventually needs to be done, and I'm planning to do it. I never got a chance really to do that, because having cancer is important, but then it comes which cancer to use, right? Which treatment to use with this cancer. So I think having cancer in the mouse with those chemotherapy treatments and testing inhibitors could add sort of additional confounding factors for data interpretation initially. So I think once we have a target and maybe a good strategy, I think then is the time to test different mouse models with different types of cancer and with different types of therapies to see how this impact cancer and mouse fertility as well. Take it as it was very clear and you don't have any questions.
It's very great. So Ariella, if we don't have more questions from the participant, probably we can end up from here, right? Oh,
Yep,
Mahmoud,
I think
can you
so.
hear us?
Yeah, thank you so much, Ewelina. It was amazing. Thank you. Yeah.
And if
We'll
you
see
have
next
questions,
month.
just email me. I can answer by email.
Absolutely. That
All right.
sounds
Thank you
Thank
for
you.
having me. Bye bye.
good.
Sure. Have a great day. Bye.